Good morning everyone. Welcome to all 4 e-learning classes. This is Zoology session 7 third chapter. So let's have some quick look of the remainder of what we have seen yesterday. That is ABO blood grouping system. A is the blood group. There are 4 phenotypes, 3 alleles and 6 genotypes. A is a blood group which is with the A antigen in its RBC and the B antibody in its blood plasma. In the same manner, the B blood group is with the B antigen and it is with the A antibody. AB blood group is with the AB antigen and no antibody, whereas O blood group is with no antigen and it is with the A and B antibody. Next, generally these blood grouping system in which AB is AB plus AB positive is said to be the universal recipient. We have discussed universal recipient. The AB positive is considered to be the universal recipient. As I already told you that the blood grouping system in which besides the ABO, one more is there which is none other than the RH blood grouping. In the year of 1940, Landsteiner and Wiener are the 2 scientists who identified the another factor which is RH factor, or it is also named to be the RH blood group. So whoever the individual have ABO is compulsory. A blood group, B blood group, AB blood group and O. So here AB positive in the sense the individuals that are with the RH antigen are said to be the positive. I told you that genetic incompatibility in the sense the inability of coexisting of the inability of the existing of 2 different blood groups together is what named to be the genetic incompatibility, which means if you donate a blood group of one kind, the recipient if it is another type, that is what named to be results in genetic incompatibility, where if A blood group is given to B, so that is genetic incompatibility. Here particularly positive and negative will be of more importance to be discussed. So if a positive blood donated to the negative individuals, it results in genetic incompatibility leading to the rupturing of the blood vessels. That's the reason why the marriage between positive husband and negative wife is not suggestible. So that's what so positive is always going to stimulate the production of antibodies in the recipient. Positive in the sense RH antigens, most of the population are with the RH antigens, and RH antibodies do not exist naturally. So if a positive blood group transfused to the negative individual, in the negative individual which are not with the RH antigens, so they sensitize and start developing RH antibodies. So RH antigen and RH antibodies clump, agglutinize, results in the rupture of the RBCs, which lead to the fetal. So blood transfusion and the pregnancy time, this genetic incompatibility going to be the vital role, critical role. So now, so it is correct to transfuse the negative blood, and it is correct to the donor when it is a negative, the recipient should be positive. In majority cases, in all cases. So AB positive is universal recipient. That is, they are positive. So if recipient is positive, there is no genetic incompatibility existence. Why? Rh antibodies are not synthesized in a positive individual. I told you, when positive blood donated to the negative blood, in the negative individuals, the Rh antibodies are produced, synthesized by sensitization of immune system. So, if the individual recipient is positive, then there won't be any problem. That's the reason why AB positive is universal recipient. In the same manner, the individual with no antigen is a negative. O blood group, it is to be particularly mentioned whether it is positive or negative. When AB positive is a universal recipient, correlatively O negative is the universal donor. Why? O doesn't have any antigens naturally like that of the A antigen and B antigen. Besides that, they are also not with the Rh antigens. So when they donate, so they are not going to stimulate or produce the stimulation of the production of the Rh antibodies in the recipient. So O negative is a universal donor. Rh blood group, the second one, the another type we discuss is a Rh blood group. Generally, Rh blood group, its history is in the year of 1940, Landsteiner and Wiener are the two scientists who have first identified one more besides the ABO. That is another besides the ABO is none other than the Rh blood group. Its discovery was done in 1940 by Steiner and Landsteiner and Wiener while working with the rhesus monkey whose the name is Macaca mulata. Macaca mulata is a rhesus monkey. That is Macaca mulata, in which while working with this blood transfusion experiment, one more factor antigen identified, which is named to be the Rh factor or Rh antigen. Rh factor, Rh antigen. In the most instances, it is also named to be the D antigen. It is also considered to be the D antigen. The persons that are with the Rh antigen are what considered to be the Rh positive. The individuals that are with the Rh positive, Rh antigens are said to be the Rh positive. I repeat, the individuals that are with the Rh antigens, basically the antigen is a dominant. Basically a gene that has a factor, a gene what produces an antigen. So here Rh antigen, such are what name it to be the dominant individual. A dominant is a positive. It can be in two status. One is named to be Rh positive homozygous and another is the heterozygous. The individual, these are with the Rh antigen. The individual who are not with the Rh antigen is denoted as Rh negative, is Rh minus. It is what we say is, in order to express you, in order to give you the explanation, the double recessive is nothing but negative. Double recessive status. Basically, the dominant factor is every gene has the alleles. So in this connection, the dominant factor is capital R, and the recessive factor is smaller. So when what type of the genotype, dominant, recessive, you can see that is capital R homozygous, and the capital R smaller heterozygous. Dominant will be of two types, and recessive is a double recessive, smaller, and its nomenclature is also, now we'll see. So the individuals that are with the Rh antigen are what said to be the Rh positive. Those who do not have Rh antigen, D antigen, it is named to be the Rh negative. Majority of the population are with Rh positive. There is a variation in terms of the Rh positive, presence of the Rh factor among the population, depending upon the race. The white individuals, what I am saying is, depending upon the race, the Rh is identified. Generally, all over the world, the white are Rh positive and Rh negative. Let's see how many are. What is the condition of the Rh positive, Rh negative status? Depending upon the race, whitish population, what I am saying is white. 85% of the whitish population are with Rh positive, whereas 15% is negative, Rh negative. And if you see American black, which means in America also you do find the black. American blacks are 95% or Rh positive, whereas Rh negative is 5%. In the case of African Negroes, African blacks, or the Negroes who are 100% Rh positive. So what you come to know is, depending upon the race, the Rh condition can be explained. That is, whitish population, among entire world, whites are with 85% Rh positive, whereas 15% is Rh negative. In the same manner, American black, in America, the black individuals are 95% are Rh positive, and the 5% are Rh negative, whereas African Negroes blacks are 100% Rh positive. What we come to know is majority of the population. Whenever if you are in a classroom, if you find out the total number of the students, if you go and calculate 95%, so it varies from 85 to 99 among the white, whereas in the case of the Negroes, it is 100%. What I am saying is, entire the population of the world over, majority are Rh positive, means they have Rh antigen, D antigen. Such are what name it to be the Rh positive. Basically, the dominant factor is capital R, whereas the recessive is smaller. Generally, the Rh positive and Rh negative individuals are phenotypically normal. What do you mean by this? I repeat, what I am saying is, Rh positive and Rh negative individuals are phenotypically normal. Then why we are discussing this as a topic? During the blood transfusion and the pregnancy time, the Rh blood grouping test is very, very important. One more point to remember. What I am saying is, Rh positive and Rh negative individuals are phenotypically normal, means they do not show any harm symptoms. But during the time of the blood transfusion and pregnancy, it is to be taken into consideration that the blood is to be tested. Donor should be negative. So, donating blood is required more. Required is more. So, what is the consideration to be done? That is recipient. If the recipient is negative, then majority are positive. So, this leads to genetic incompatibility. So, probability is if the recipient, if it is positive, just now we have seen AB positive is a universal recipient. So, if the recipient is positive, even if the donor is negative, it does not cause any harmful effects. So, while blood transfusion and pregnancy, it is what taken into consideration, that is whether it is going to be showing the effects, harmful effects. If the recipient is the positive, as majority of the population are positive, so it is not going to be showing any the harmful consequences. If that's the reason why while pregnancy, a woman when she is pregnant, definitely by the 7th month and early also the blood test is done. If she found to be RH negative, then the baby are going to be prone for HDN or HDNB, erythroblastosis fetalis, or hemolytic disorder of newborn. So, the genetic incompatibility exists when donor is positive and recipient is negative. So, blood should be tested. If the recipient is positive, not at all problem. Why? Generally, if the recipient is negative, what we explained earlier, we have discussed that RH antibodies do not exist in the nature. Majority of the population are with RH antigens, nana. Whereas no population will have RH antibodies naturally, as that of we have seen the A antigen and B antigens are naturally existing, and A antibody and B antibodies are all not also existing. But RH antibodies do not exist. So, as RH antibodies are not existing naturally, RH antibodies are produced when positive blood injected to the negative individual. While blood transfusion, if the positive blood given to the negative individual, there is a chance of production of RH antibodies. But if the donor is RH negative, the individual is recipient is positive, it is not going to be showing any ill effects or consequences in the recipient. So, it is the thing to be taken into consideration that the recipient should be positive. If the recipient is positive, no consequences, harmful consequences are going to be happen in the progeny to come and also the recipient individual. So, RH positive, RH negative, both the individuals are normally phenotypically normal. They are phenotypically normal. They do not show any the symptoms of the harmful effects. Whereas transfusion and the pregnancy time, it is going to show the harmful effects. Pause. Now, so RH antigens are naturally existing. Majority of the populations are with RH antigens, so they are RH positive. Naturally, RH antibodies do not exist. They are produced when RH positive blood transfused into the RH negative individuals, or else when there is a marriage between a RH positive husband and the Rh negative wife, the first baby is going to be Rh positive, and this antigen is going to be stimulate the mother immune system and Rh antibodies are produced. That's the reason normally the Rh positive, Rh negative individual phenotypically normal, whereas the blood transfusion and the pregnancy time, it shows the harmful ill effects. We have discussed that the ABO blood grouping system is under the control of the gene which is I, and it is present on the chromosome 9. In the same manner, Rh antigen controlling alleles are, Rh antigens alleles are present on homologous pair of chromosome 1, homologous pair, chromosome number 1, homologous pair, chromosome 1. So, like that of the ABO blood grouping system, this is also a autosomal trait. That is, the genes regulating the ABO blood grouping system and Rh antigen system, Rh blood group system is under the control of autosome. ABO is controlled by the genes present on the chromosome 9, and here the chromosome 1, homologous pair of chromosome, in the sense homologous in the sense the genes present on a chromosome. Naturally, chromosomes are inherent vehicle which contain certain places or locations, name it to be the loci or locus, upon which the genes are present. So, the chromosomes that are structurally, functionally, behaviorally same are named to be the homologous pair of chromosome. Homo mean refer to the same. So, homologous pair of chromosome 1 where the alleles, the genes that are responsible for Rh antigen are present. Based on this, we go for the nomenclature of Rh blood grouping system. Nomenclature, there is two types of nomenclature we came across. Two types of nomenclature. One is Fisher and Race nomenclature system, whereas the another is said to be the Wiener nomenclature system. What do you mean by this? That is the inheritance of Rh antigen, how takes place? What is the process involved? What are the alleles? Where do they locate? Is what we are going to discuss here. What we have discussed in the case of the ABO blood grouping system. In ABO blood grouping system, the gene responsible for antigen A, antigen B, or none other than the allele IA, IB, and the one that are do not have antigen are with the allele IO or recessive I. All these three alleles are present on among the population. I am saying all the three alleles are present in a one loci. That is, the genes with the two alleles are present on a single, that is present on chromosome pair, chromosome 9th. That is a single loci. Whereas here, out of the three, any two, IA, IO, IA, IB, IO, likewise 6 genotypes we have seen. In this connection, unlike the ABO blood grouping system, according to Fisher-Race, the chromosome pair, homologous pair of chromosome 1, look at here, this is the pair of chromosomes which have the three loci 3 loci are present in each of the chromosome pair 1 in this connection of RH antigen. And they have the 3 pairs of the genes and each gene have 2 alleles. Is it right? That's what it is said to be the CDE nomenclature system. According to Fisher and Race, there are 3 pairs of genes present on 3 different loci, where they have a pair of chromosome number 1. Chromosome pair 1 have 3 different loci where there are 3 pairs of alleles. We see each gene have a pair of allele. Look at here. It may be with capital C or small c. I repeat, it is either capital C or small c, capital D or small d, capital E or small e. The same way another, either capital C or small c, capital D or small d, capital E or small e. This is what the CDE nomenclature, which means as we have seen IA, IB, IO, here the CDE are the alleles which are going to be playing role in the nomenclature of Fisher and Race, which means this is the chromosome which have what type of genotype? 3 alleles. It may be C, D, E. Look at here. Understand? What I am saying is a pair of alleles in which it may be C, D, E, capital C, D, capital E, or else it may be small c, small d, small e, small c, small d, small e. That is, this chromosome may have either this combination or this combination. And apart from that, it may be capital C, capital D, small e, capital C, capital D, small e, or else capital C, small d, capital E, capital C, small d, capital E. All possibilities what we refer here, capital C, capital D, capital E, or small c, small d, small e. In the same manner, capital C, capital D, small e. All possibilities we can see capital C, small d, small e, capital C, small d, small e, capital C, capital small d and small e. Likewise, you can write as many. That is, all possibles we can here can see capital C, capital D, capital E, small c, small d, small e. So each chromosome is denoted by means of this genotype. Capital C, capital D, capital E, small c, small d, small e. Next, the same another is also the homologous pair of chromosome. What one have, the same is also with the other one. Second probability, capital C, capital D, small e, capital C, capital D, small e, or else capital C, small d and capital E. Next, capital C, small d, capital E, or else even you can see that is capital C, small d, small e, and the here like this also you can see. So these are all what probabilities that are existing. And the point to remember, presence of capital D allele, presence of capital D allele is going to be considered a person is a positive. Earlier we have seen that is a dominant allele is going to be considered to be positive. So according to Fisher and Race, presence of a capital D allele is going to be, for suppose here this is capital C, capital D, capital E, small c, small d, small e. Here capital C, capital D, small e. Here capital C, small d, capital E. Here capital C, small d, small e, capital D, C, small d, capital E. In all in these instances, presence of capital D is going to be determining an individual positive. So in this pair of the chromosome, one capital D is there. So capital D small d here, here capital D small d, here small d small d, which means these two individuals with the chromosomal status are Rh positive. So what I am explaining is presence of capital D will determine an individual Rh positive, and here this individual is recessive. Why? A dominant can be homozygous dominant or heterozygous dominant. Look at here, these two individuals are with individuals in the sense here the pair of chromosomes, here capital D is there, capital D is there. So presence of capital D determines an individual positive or Rh positive according to Fisher and Race, and here the double recessive is a what is called Rh negative.